talk about my PhD, but also a bit about work that I did before that. And I will focus on one particular project. But first, I think it's important to say where it all started for me. So in 2015, I was working with a large community on mycelium materials. So initially I was trained as an architect. I have a diploma as an architect, and obviously without any background in anything which has to do with biology. So that's why I was trained actually in all the lab techniques by fellow citizen scientists. And so I quickly also started to train other people with the knowledge that I learned through them. And along the way, we built our own lab, as you can see here in the pictures above. And we started to train a lot of people, children, teachers, designers, with the knowledge that we gained on mycelium materials. Most of this knowledge has been published online in open source manuals, for example, on the BioFab forum. We were doing 99% of this work voluntarily. So it's also important to say that most of the work was actually just in our free time. We also, like Dylan already said, founded the company Flimspin Bio, which is .bio, which is based in Belgium, and which at this point evolved towards a consultancy-based company in biodesign and circular economy. And then I ended up starting a PhD at the University of Brussels in Belgium. And it's really an interdisciplinary research. So it united for the first time different faculties at the university, which is the group of microbiology, physical chemistry and material sciences, and the architectural engineering group. And if you want to download this PhD dissertation, you can just look for the title on Google or on ResearchGate and download the full PDF. It's and during the time I was doing my PhD, I was very fortunate to be able to work with brilliant master students from different disciplines, because they have taught me their experience with microbiology, with mechanical engineering, material sciences. And so by engaging those affinities and entanglements with those multiple disciplines and along also the different scales and the different language that those disciplines have, it's kind of allowed me to better understand the sophisticated capacities of biohybrids materials and of course about mycelium. So yeah, I hoped that the work could lead or could inspire to also the acceleration of the debate about how to radically rethink materiality in a sustainable way, but also in a social way. But yeah, anyway, we will end up with the earth that we deserve. So with the research, I wanted to understand what it takes to valorize organic waste streams from wood and agricultural industries while creating the solid material that is biodegradable at the end of its life cycle and that has been made with an abundant glue coming from fungi. And so here the video you see was a collaboration with different Danish companies where we had the goal to test the scaling of the manufacturing. And so the main goal of the PhD was to explore the principal factors that affect the biological and the material properties of mycelium materials. So that's what we did in the first, what I did in the let's say the first chapters of the research. But then it's also a hybrid investigation across different disciplines that aim at contributing to the field of the manufacturing process with mycelium. And so today, I will talk about one small aspect of the research, which is focusing on scaling the fabrication of mycelium materials through robotic fabrication. I approached this step by step, first by starting to understand the properties of mycelium materials themselves, and then trying to understand the requirements of the organism to stay alive during a longer lifespan. So one of the problems that we are facing when we are growing mycelium materials in molds is that there is a lack of air, and this will significantly restrict the viability of the fungal organism. So the mycelium is really unable to grow on the bottom because of the lack of oxygen there. And actually molds also restrict the complexity in terms of the final product. In terms of geometry and scale as well. So that's why I wanted to understand to what extent the implementation of digital tools could facilitate the large-scale applications, and whether the living organism would also survive those harsh conditions in which it is subjected. So the first purpose was to understand whether pre-growing and mixing the material during the growth of the organism would impact the mechanical properties. So the idea here was that the substrate is grown, pre-grown in bags, would stimulate a more high-flow robustness, because we could kind of knead it and massage it. And that would also allow more airflow within the substrate. But then on the other hand, if you grow the substrate directly in the boxes, the density would also enhance, because you can compress the fibers more densely. So those two things were tested here. And from the bending test that we did with pre-grown, with different types of samples here. So we have pre-grown samples in the bags, and samples that were made directly in the molds. And then I also tested different compression, densification forces on these materials. So you can see that the mechanical properties enhance quite clearly when samples are pre-grown, which was kind of expected, but it's always nice to see it in a graph. And so before the fabrication of the large mycelium blocks, I did some experimental work to select the most appropriate growth protocol, because it could be approached from different ways, and I knew that mycelium materials wouldn't really grow through the substrate if the samples were much larger than about 10 to 15 centimeters. So I tried different ways. So first by growing everything in boxes in advance, and then placing those blocks horizontally next to each other in a larger panel. I also tried to stack compressed blocks vertically, and also stacked pre-grown but not compressed blocks. I will quickly go to all those experiments. So here the samples that were grown directly in the boxes and then placed next to each other didn't really achieve a seamless connection between the blocks, and that's actually along the whole research was kind of a challenge to have this seamless connection. Then with the second test was performed by first cold pressing all those living blocks before stacking them. And the goal here was to, like I said before, to enhance the mechanical properties by having a larger densification. But if you compress blocks that are still alive and still wet, the water that has accumulated in the samples will just come out, and the outer skin of the material was damaged. And also during this procedure, it was very clear that the core of the material wasn't really holding the material together. Because you can see that, yeah, fragments of the initial block just broke off. So these results weren't very homogeneous. And then I also tested to stack different blocks on top of each other. These blocks were pre-grown, placed in a mold, and then stacked on top of each other, but without the compression phase. And that was more, a more successful approach to reach those bigger blocks. And then different types of fibers were also tested. Different species were tested. So here we see the growth on hemp fibers and on beechwood fibers, where the beechwood fibers would be colonized a little bit faster than the hemp fibers. And different species, so this is Hynoderma lucidium, and this is Trametes versicolor. You clearly see a difference in growth behavior. So in the end, I decided to go for, to work with Hynoderma lucidium. Okay. So where are we? Yeah, so I then moved on to a bigger scale by having those multiple growth phase methods. So 2 blocks were made on a larger scale. One block was dried before the robotic process, and then the other one was left alive during the whole process in order to allow this biological welding at the end. So during the first growth phase, the mycelium was grown in bags, so you can see here. And those bags all contain this filter system, which allowed the airflow. And then the mycelium homogeneously colonized the whole substrate, and almost every day I kind of kneaded the substrate to make sure that it would distribute everywhere in the fibers, which then also allowed a bit more robustness. And so after 8 days of growing in those bags, the substrate was then crumbled by hand, and it was distributed in those plywood molds that could be reused. And then the top of the mold was covered with breathable foil, and it was again incubated under those conditions. And then after, so the conditions were regulated temperature at 26 degrees. And yeah, and that's it. And so after 3 days, the substrate had taken the shape of the molds. The reason why it's only 3 days here is because it had already pre-grown in the bags in advance. And so this was enough to remove the samples from the molds. And then the panels were, the different layers were stacked on top of each other and then incubated again for another 4 days. So during this third growth phase, the different layers grew into each other. And one of the blocks was then dried in a convection oven at 70 degrees during 2 days until the weight had completely stabilized and all the humidity had evaporated. And then the other block was not dried. It was just kept humid. I wrapped it in foil. And I often receive the question, yeah, why did I choose those dimensions? Well, I think it's obvious from the image. It had to fit in the oven that we have in our lab. And so this resulted in two monolithic mycelium blocks. And then I shipped the 2 blocks from the lab in Belgium to Denmark. And luckily for me, the post office didn't open my box because I'm not sure if they would have understood what was in there, like a smelly mycelium whitish thing. And also if it would have been open, I'm afraid it wouldn't arrive properly. But so the reason why it was shipped to Denmark is because I had the possibility to use this large robot. We worked with the Danish company Odico. And so here a wire is turning at a high speed around the frame. And this way we can cut different materials into any shape we want. So this resulted in a curved shape in order to use this block as a formwork. So usually they use EPS foam for this type of applications. And the goal here was to test whether we could replace EPS foams, which also comes in those cubical forms, whether we could replace it with mycelium materials to use it, for example, for different, it can be used for different functionalities, like a formwork for concrete, for example. So here, I shipped those back to Belgium, and then I applied a beeswax directly on the surface, and this was to avoid the absorption of the poured concrete. So concrete would be poured in between here a 10 centimeter layer of concrete was poured into this. And so possible applications of this could be, for example, prefabricated structural, thermal or acoustic insulation blocks, or components that would, for example, be placed in the soil and that should biodegrade after the concrete the concrete foundation would be poured, for example. But the thing is that by drying the mycelium material, the organism is heat killed, and therefore it's also removed from some crucial biological functionalities, such as its capacity to regenerate itself. And so that's why I further wanted to investigate the potential of keeping mycelium alive during the entire fabrication process. So after the wire cutting process, the blocks were again sent back to Belgium, and the 2 pieces of the living block that you can see on the left image, they were reassembled. And then while the organism continued to grow, it produced a dense white grayish skin around all the exposed fibers, which you can see in the right image. And so during that phase, that fourth incubation phase, actually, the humidity was increased a lot until 90%, which allowed the HEFA to also bridge the gap of about 5 millimeters between those 2 elements. And so the two pieces were reconnected. And I think this small prototype demonstrates the potential of using the living organism, which had survived the uncontrollable environment of the transport and the completely unsterile environment of the wire cutting facility. It had also been damaged. So by cutting off the sizes and cutting the curved shape, it had to regenerate itself. Of course, there are many challenges to keep mycelium alive. For example, in the right image, you can see a little bit that the organism starts to differentiate to create fruiting bodies. Obviously, we don't really want this in building materials. So as the organism continued to grow, it also produced a denser network. You can see on the left image, after 30 days, this is a microscopical image. It has a much denser mycelium network. But the other challenge is that when mycelium composite continue to grow, the properties, mechanical properties will decrease over time because the organism continues to degrade the natural fibers. And this was also tested mechanically. So after 10 days, 20 and 30 and 40 days, you can see that the compression strength actually diminished. I think we might talk about the advantages and limitations of this project. If you haven't seen it already, it has been published, so you can read more in detail the whole process, and especially the discussion where I continue to explain the advantage and disadvantage of this methodology. So yeah, this being said, I'm at the end of my presentation, and I would be happy to take questions afterwards.
So the title of my presentation is From Waste to Biodegradable Architectures with Local Fungi Species. But I'll be talking about a couple of different things that we're doing at like now and we did before. Let me see if I can. Okay, so as Dylan said, I'm an assistant professor of architecture at Penn State, and I have founded the Format Matter Lab. It's located within the Stockman Center for Design Computing at Penn State, and I have PhD students, master's students, and some undergrads working with me in the lab. In the works that we do, we explore making and material exploration as integral parts of the design process. And whether it's carried out by digital tools or by hand, we approach making as part of a computational process. So we follow the approach to computing as a way of thought and reasoning, and not necessarily with the use of digital or computers. And we acknowledge the importance of like sensory engagement with and the uncertainties brought by the material things in design reasoning and creativity. And in such an approach, materials, tools, form, and geometry are strictly independent, and they are interdependent, and there are things that you can do with one fabrication tool that you cannot do with another, or there are things that you can do with one material that doesn't work with another. So we are exploring these things. We worked with clay printing before, and like different materials, different fabrication techniques. And since 2018, I believe, we are exploring mycelium. And okay, wait, how do I go? Okay, so a previous paper that I've published back in 2015 from my PhD dissertation outlines the focus of the lab. In that paper and also in the PhD dissertation, I've made a distinction between two modes of making, as making of and making for. in making of, and by looking into the interrelations between ideation, representation, and materialization and design. In making of, the focus is the ideas are conceived in representational media such as drawings and digital models, and materialization involves processing of the information available in these representations. And within this framework, materialization does not necessarily inform the design process, and the focus is mostly on the seamless connection between design representations and their materialized outcomes. Whereas in making for framework, making shapes design ideas, and the designer also values the exploration realized through making. So rather than the end product, the focus is on the improvisational process and the unexpected discoveries that occur due to the properties of materials, or and techniques that are unforeseen in abstract representations such as drawings or digital models. So there is in our design processes, we go between making ofs and making fors, and there's an alternating dialogue of control and uncertainty. And I think this also can be like the uncertainties in making for can be computed. There were similar approaches to these kinds of distinctions. David Pye, in his famous article, he distinguishes between workmanship of certainty and workmanship of risk. Tim Ingold, also, who's an anthropologist, differentiates what he calls the standard view of making, which is closer to the making of, with what he calls the textility of making, in which he suggests replacing the standard view with, again, I quote from him, an ontology that assigns primacy to the processes of formation as against their final products, and the flows and transformations of materials as against states of matter. So, I mean, even we probably have seen in the previous talks as part of these talk series, and also in Elise's talk, like in Mycelium, when working with Mycelium, it's definitely a making for process. There are so many uncertainties, and then you have to constantly think about the final form, but also but by getting informed from the material itself. Okay, so about Format Lab, so I don't do anything alone. I always work with an amazing group of students, and this is the current group who work with me. The top row is the Mycelium group, and Ali, who presented in the previous talk series, is a PhD candidate. We started this Mycelium research together. He's exploring the structural aspects of mycelium-based composites. Natalie joined us last year. She's currently an undergrad student, but she is starting a master's program also at Penn State, and she's going to be working also with Mycelium. And Alale joined us this semester. Natalie is working on exploring acoustic properties of Mycelium, and Alale will be exploring robotic printing of Mycelium. So, okay, a little bit of history. I teach a course called Hacking Materials and Production Methods, and this is how my interest in mycelium started. In this course, the conventional teaching structure in which, the instructor is the big circle and the students are the small circle is challenged. And sometimes, I become the small circle and the students become the large circle. And every semester when I teach this course, I come up with different things to explore. So the things that we do in the course is also new to me, are also always new to me. We built several machines, clay printers, CNC machines, and in one semester, fall 2018, I was curious to explore, because I've never worked with living materials before, I was curious to explore how we could design and work with living things. So in this class, I separated students into several groups, and one group was working with mycelium, was tasked to work with mycelium, and I've never worked with mycelium before myself. There were other groups who were working with bioluminescent algae, some microbes in the soil, and some other stuff. But for this presentation, I'm going to briefly talk about what they did. So 2 students in that class, Andrew and Tyler, after making lots of hands-on explorations with mycelium that they have grown themselves, but also that we wrote innovative kits for them to begin, but they got so excited and they, went on want to grow mycelium themselves. The project that they developed, or the conceptual project that they developed, was to make blocks with mycelium, but also use mycelium as the mortar between the blocks. And we just saw in Elise's presentation that that's pretty much possible. So they made some tests exploring, using mycelium as the mortar between the blocks, but also they on purpose impaired the block itself, and then used mycelium and explored to see if mycelium can repair itself by growing in between the different parts of the block. So this is actually how it all started. And they also presented their explorations in a symposium in Lisbon. So that was also their first experience going and presenting as undergrads something in an academic symposium, and they enjoyed this. So then, I got interested on mycelium specifically, also because at Penn State there's a mushroom research center, and then so I got in touch with them, and at the same time, Ali was starting his PhD, so we decided to, and his background was in civil engineering, so we decided to see if he could explore the structural aspects of mycelium-based composites. So in 2019, we started our collaboration with my Mushroom Research Center at Penn State, and then with some developing different composite materials, and then testing how they perform structurally, just to get a little bit used to working with mycelium. So fast forward this year, and this is part of, again, Ali's PhD dissertation, but there's a large group behind it. We built our first load-bearing structure called Microprint. It's a large team. The top row, again, is the group who is behind the design and the the main design of the structure. And then while we were assembling and fabricating the components and assembling the structure, there were lots of people who were helping us, as you can see in this picture. And also at the bottom are our mycologists who helped us and then provided us rooms for growth and insights for mycelium cultivation. So a little bit background: Arman and Ismail are two collaborators in this research. They're from Iran, they're not from Penn State, and they have previously built a structure. This is a structure called Concreate, using concrete. And then as you may know, mycelium-based composites work best under compression, and then they are very weak in tensile strength. So the form finding of the concrete that they developed was suitable for an exploration also with mycelium, because the structure works under compression. So we worked with them. We made an application for the IASS Innovative Lightweight Spatial Structures competition. And then we started working towards that competition entry. Again, like I said in the beginning of the presentation, in working with biomaterials, mycelium, materials geometry, and the fabrication techniques used are interdependent. So we started exploring we started working towards this competition entry, and the first stage was material cultivation. So this is, the basic mycelium-based growth chart. It starts with inoculation, and then you first prepare the substrate, mix them, sterilize the substrate, let them cool down, and then inoculate with fungi. For this project, we used sawdust and straw, and then inoculated with the locally abundant oyster mushroom species. Even now, in Pennsylvania, it's very abundant. It's a very abundant species. Our yard is now infested, I would say, with oyster mushrooms. And then during the growth stage, we first grew them in bags, and then we created formworks to grow the components. And then there are multiple ways of seizing growth to obtain the mycelium-based composites. In our case, we dried the components in open air. And then, but an alternative would be to heat it, which would kill the mycelium. So material tests, this is part of Elise, also PhD dissertation. He developed 35 different samples and tested their compressive strength. And the samples, the difference between these samples was the sawdust to straw ratio. That was one thing. But another difference was how long they grew in bags versus how long they grew in formworks. So there were like multiple parameters. And then comparing all these samples under compressive strength tests, we identified 4 composite mixtures that performed the best in terms of compressive strength. And then we decided to use these 4 different composites in the design of the structure in different parts, depending on where we need heavier and stronger materials, and then towards the upper parts of the structure we would need lighter materials. So based on this, with the help again with our collaborations with Arman and Ismail, we did an initial form finding, and then let me see if this will play, to design to like propose for the competition before building the structure. And then here's a video showing how the form is generated and then how it's discretized into smaller components to enable also for ease of fabrication, but also to enable mycelium to grow better within the formworks. Okay, let me see. So using the same form finding method, that could be, you can generate a variety of different forms. So after that, we started making several prototypes just to see, just to test mycelium growth and make some changes in the formwork design. And we faced several challenges because the scale of these was pretty big, and then we kept updating the formwork design. So first we were using chipboard as materials for the end plates, as you can see over here. And then mycelium growth was okay, but we had like gaps towards the end parts, and then some cracks in the joints. So we kept, I didn't include all of the prototypes that we did in this presentation, but we changed the materials, we explored different materials. So first we were using plastic to make the shells of the formworks. Then we changed those with paper-based materials, because within plastic, mycelium was not able to breathe, and then completely. And then we changed that with a paper-based shell material, which is also like recyclable. And then we started our new sets of prototypes. This is one of the structures, small scale structures, prototypes that we built before building the large scale competition entry. But I'm going to also talk about the challenges we faced later. So after making the prototypes, learning about the size constraints for like better mycelium growth, but also like how to fill the formworks, how to best fill the formworks, how to best compress the substrates within the formworks. Based on these, we made some changes in the formwork design, and then these informed the final form finding, which was simpler than our initial form, and that the initial form that we submitted for the competition entry. Okay, so it was made out of 64 components, and then this is, each of those formworks had a interior skeleton, so this is how it looks. And then based on our the different composites cultivated, we distributed those composites this way along the structure. So the first pieces at the bottom were made out of aluminum, just to make sure that the mycelium does not touch the soil. And then the heavier pieces and the stronger, denser pieces were the ones that are right after the legs. And then as you go up, as you move up, the structure got lighter. This is a schematic drawing of one of the complex components. And then the blues and the red parts that you see, the end plates and what we call the guide straps, we explored multiple materials, and then for the final prototype, we decided to use birch wood, which was a bad choice. Okay, so fabrication and assembly. The mycelium grew at Mushroom Research Center in controlled environments for 10 to 14 days in bags. And then in the meantime, we fabricated the formworks using laser cutter and water jet cutter for the aluminum pieces. And then we filled the formworks by hand, and then they grew within the formworks for another 7 to 10 days. And finally, we dried the components. So a couple of challenges we had was that because we had a deadline for the competition, the components did not fully dry by the time, and it was very humid at that time. Okay, so here are some of the components of the structure that we were going to assemble, and you can see that there was not enough growth, and then they were not fully dry. And here we are assembling the structure. Since it's a compression, since it was fully under compression until we placed the last piece, the structure was very unstable. So that's why we're using like like whatever we could find to support the structure. Okay, here's a time-lapse video of the assembly process. Okay, and this is after we took out the support stools and other stuff. Okay, and okay, up, why? I'm trying to, okay. So the structure, unfortunately, collapsed later that night, and there was also a storm. So we were working under tents, and it was a little bit challenging. But the main reason that the structure collapsed was because we had contamination in some of the components, and which made the growth of mycelium, which like prohibited the growth of mycelium, and then the contamination, like during when they were drying, spread also to the other components. So that was a problem we faced. And then the other problem was that we didn't have enough time to dry and grow the components because of the deadline of the competition. So contamination, we think, was mostly because of the wood that we were using. Some of the end plates, we were not able to sterilize them properly. And then since, it's a structure made out of too many components, that increases the chances of contamination. And then trichoderma, is very resilient, so it spreads easily. So that was one of the challenges we faced. And then, but our goal is to, since after building and assembling the structure, it held its form. That informed us that the form finding was nicely done. So our plan is to rebuild it by revisiting our growth protocols and also like the materials that we use in the formworks this semester. Okay, another project that is that we're working on is Microprint, but it's currently, like, we're working on this now. And then in Microprint, we are using waste cardboard and waste paper as we're exploring the use of waste cardboard and waste paper as substrates. We're again using oyster mushroom for inoculation, and then our goal is to integrate advanced like augmented robotic printing to the process. We did some quick tests to see how the mycelium grows. And then Natalie is a student who's working with me. Like I said, she's exploring the acoustic properties of mycelium that she grows on paper and cardboard and newspaper waste. So this is newspaper. The greens are not mold, but newspaper. So our goal, and another PhD student of mine who's not working with mycelium is working, and together we are working on this project called C-Sense Respond, Augmenting Robotic Fabrication. The goal in this project is to make the robots, increase the awareness of the robots from their surroundings. So currently, without any augmented capabilities, robots don't don't know about what's happening in their immediate work environment, and they also cannot capture failing material indeterminacies. So what we are working on is to develop accurate 3D capture and to develop frameworks to accurately 3D capture and reconstruct the work environment so that you can actually work on any type of surface. It doesn't have to be a planar surface that you have pre-modeled in the computer. Develop adaptive toolpaths, frameworks that can work on scanned data, and then integrate additive manufacturing tools that can print on any type of surface, and do this later on in real time. So our goal is to integrate the C-Sense Respond framework, robotic fabrication framework, and microprint that will allow us to fabricate more complex forms and to print mycelium-based composites on also non-planar surfaces. So this is something that we are working on. Okay, I think, yep. So, and then for that, we are exploring the use of waste cardboard, waste paper as substrates so that it could, again, contribute to a circular economy where we use waste cardboard, 3D print, construct things, and then, again, crush it, compost it so that it could, again, later on be produced as paper or cardboard materials. Okay, I think this is all from me, and I'd be happy to have your questions. And then if you want to reach out, this is my email. And thank you.